see that. So thanks very much, Philippa, for joining us today. It'll be great to hear about the work you're doing. Cheers.
Okay, well, thank you for the opportunity to talk to everyone today. Thanks for coming. So hopefully you can all see my slides. I presume Geraint will tell me if not. But today I'm going to talk about wheat genomics and specifically about what work we've been doing to try to understand gene expression in polyploid wheat. So the reason that I'm so interested in polyploidy is that polyploid plants are abundant around the world. So over 70% of flowering plants that grow in the natural world have a polyploid evolutionary history. And it's not just natural species, it's also major crop plants. So you can see a selection of polyploid crop plants on the slide below. So things like oilseed rape, cotton, potatoes, bananas, sugarcane, and wheat, my favorite, that you'll be hearing a lot more about today. And these polyploid genomes make them have very complicated genomes with multiple copies of each gene, which poses a challenge for crop improvement. Because you need to know which copies of the genes, or all of them, you need to target to improve specific traits. So wheat became a polyploid through its evolutionary history by first a hybridization event between the A genome progenitor Triticum murata and the B genome progenitor, which is currently thought to be extinct, but something similar to Agelops speltoides. And this hybridization event took place about 400,000 years ago to make tetraploid wild Emma. And this wild Emma underwent a second hybridization event with the D genome progenitor Agelops tauschii about 10,000 years ago to form hexaploid wheat, hexaploid bread wheat that's grown around the world today and is 95% of the wheat crop that's grown, the other 5% being tetraploid wheat. But I'll be focusing on hexaploid wheat today. So due to these several steps of hybridization, wheat has, if you like, multiple genomes within its genome. So it has an A genome, a B genome, and a D genome, which come from these original diploid grass species. And this means that it has multiple copies of many genes. And because these grass species descended from a common ancestor, they had quite similar gene content within their genomes. And not only does this make it challenging because there are multiple copies of most genes in wheat, it also is a very large genome. So just to put it in scale, these are the sizes in the same relative size scale for some other well-known species. So the wheat genome is 5 times larger than the human genome. The wheat genome 16 gigabases. And this is many multiples of times larger than the Arabidopsis and rice genomes that are other commonly studied plant species. And having this large polyploid genome poses specific challenges in wheat. So in wheat, we have 3 copies of each chromosome. So they're known as the A, B, and D genome copy. And if we have a gene that's on chromosome 7A, for example, then we'll also have a copy on 7B and a copy on 7D. And these 3 copies are likely to be very similar. So they're known as homologs. This word will come up a lot during my talk. And these homologs are over 95% similar in their coding sequences in general, which raises questions about what their functions are. Because this have such high sequence similarity, it's thought perhaps they'll have the same biological function, and therefore we might predict that these homologs will be functionally redundant. And this is a major problem because functional redundancy can hide genetic variation and phenotypic variation. And what I mean by this is if we're looking at a diploid plant and you knock out the copy of the gene that's present, you'll see an effect if that gene has a particular function. For example, if the gene's related to grain size, you knock out that gene, maybe you're going to get smaller grains. And we see some effects like this in polyploid wheat. For example, genes like the photoperiod 2 gene and vernalization 1 have dominant mutations in them, so that we can indeed see a strong mutant effect, even when only one homolog is mutated. However, for many other genes in the wheat genome, because there are multiple copies, we see dosage effects. And this means that, for example, if you knock out one copy, then you only get a small effect, so a 33% effect. Whereas you knock out 2 copies, you get a 66% effect, and then you knock out 3 copies, you get the full mutant effect. So that means that by mutating just one copy, you only get a relatively small effect size compared to in a diploid species. And an example of this is grain weight 2. And You can see here that in wheat, when you knock out 1 copy, you increase 1,000 grain weight by 6%. When you knock out 2 copies, you increase 1,000 grain weight by 11%. And when you knock out 3 copies, you get an increase of 21%. So this is really a gradual increase in 1,000 grain weight caused by knocking out increasing numbers of copies of the GW2 gene. But just for contrast, if you knock out the single copy in rice, you get an immediate large increase in 1,000 grain weight. And clearly the 49% increase is not exactly the same as the 21% increase, which really indicates we don't completely understand whether this gene has precisely the same function in rice and wheat. But the idea of this is to just illustrate that in wheat, knocking out individual copies has a more gradual effect than knocking out the only copy in a diploid species like rice. There are other possible dosage effects that we could see in wheat. For example, non-additive effects, where if you knock out 1 copy, you get a small effect. Knock out 2 copies, you get a small but slightly bigger effect. And it's only when you knock out all 3 copies, you get a really strong effect that's very phenotypically noticeable. And there are genes that are known in wheat to play a non-additive role. For example, GPC, grain protein content, and also the waxy gene involved in amylose synthesis in the grain. And there are other genes which have functionally redundant copies. So in these genes, then if you knock out 1 copy, there's no difference compared to wild type. If you knock out 2 copies, again, there's no difference compared to wild type. And only when you knock out all 3 copies do you see a strong effect on the phenotype. And examples of these types of genes include starch branching enzyme 2 and the mildew locus, MLO. And the consequence of these differences in dosage effects or dominant effects, we don't really know to what extent wheat genes fall into each of these categories. So it could be that the majority of wheat genes actually are functionally redundant. So if you knock out 1 copy, you won't see any effect. Whereas other genes, if you mutate just 1 copy, you will see a reasonable sized effect, for example, if it's an additive gene. And a consequence of this is that many of the genes that we know about in wheat and have been able to identify through QTL mapping fall into the first 2 categories, so dominant and additive effects, where we can actually see the effect if just 1 of the 3 copies is altered. Because in a QTL mapping approach, you won't be able to target, you won't be affecting all 3 homologs. And this means that really the variation that we've been able to select has been mainly focused in perhaps a small subcategory of genes in the wheat genome. And potentially there are many more genes that have important influences on phenotypes that we're at the moment unaware of their functions and potential ways that they could be used for crop improvement. And this lack of knowledge is in part because we've lacked genomic information about wheat. So until very recently, we didn't know what genes there were in the wheat genome, let alone know many of their functions. But this has really started to change. So in 2018, a fully sequenced wheat genome was published. And this was a high quality assembly with over 94% of the genome assembled. And it was assembled into pseudomolecules, so almost whole chromosome scale assembly. And importantly, there were genes annotated on this wheat genome sequence. So in total, there were 110,000 genes found in the wheat genome. This includes A genome, B genome, and D genome homologs. And as a first step towards trying to understand this question of functional redundancy of these 110,000 genes, we decided to look at RNA-seq data to try to understand the global homolog expression patterns. So for example, are most genes in the wheat genome that have 3 copies, do they all get expressed at the same level? Or is it that actually 1 of the 3 copies is more highly expressed? So we can imagine a case on the right that you can see where the B homolog is much more highly expressed than the A and the D homolog. So perhaps if we find this gene has an important trait that it influences, such as grain size, perhaps we only need to breed for changes in the B homolog because it's the most highly expressed of the 3 copies. But when we started this work, we didn't have any clear idea about whether this was going to be the case. Would the B genes, where only 1 of the 3 copies was highly expressed, or would most of the genes have a similar expression pattern of all 3 homologs? So to start looking at this question, we decided to look at the expression of genes that had 3 copies, and this was about 17,000 triads made of 51,000 homologs. So each triad has an A homolog, a B homolog, and a D homolog. And for example, if we have a triad which has an expression level in transcripts per million of the A homolog of 10, the B homolog of 10, and the D homolog of 10, we decided to normalize the total expression of the triad add up to 1. So for example, in this triad, then each of the 3 homologs has a normalized expression level of 0.33. If we take another triad, which has a different expression pattern, where the A homolog is expressed more highly at 80 transcripts per million, the B homolog at 10 transcripts per million, and the D at 10, then we can normalize this again so that we can compare these 2 triads with a very different overall expression level. And looking at triads in general across the whole wheat genome, we found that there was a higher expression level of D homologs. But although this was significant statistically, the actual effect size is very small, so that the D homologs are expressed 33.7% of transcripts, whereas in the A it's still 33.1%. So we're not sure whether this small difference is actually biologically relevant. But we also wanted to look at what happens at the individual triad level. So to do this, we plotted graphs of how triads are expressed. For example, a triad where 30% of the expression comes from A, 30% from B, and 40% from D could be plotted on the graph in this way. So A 30%, B 30%, and D 40%. And the triad is represented as a dot almost in the center of the triangle graph. Whereas a different triad, which has 21% of the expression from A, 7% from B, and 72% from D, will be plotted more towards the corner of this triangle graph, specifically towards the D corner. So we used this method to plot all the triads, the 17,000 triads in the wheat genome. And this is the pattern that emerged. So we found that 72% of triads were expressed in a balanced pattern. So all 3 homologs were expressed at quite a similar level. We also found cases where one of the 3 homologs was expressed at a lower level than the other 2 homologs. So this included 7% had A suppressed triads, 7% were B suppressed triads, and 6% were D suppressed triads. And we also found cases where one of the 3 homologs was more highly expressed than the other 2 homologs, but these were rarer, so about 2% in each of those categories. So we were quite curious, how do these triads end up with these differences in expression patterns? And our first hypothesis was that there should be something different in the promoter sequences. So for example, in the A dominant triad shown in the middle, where the A is more highly expressed than the B and the D homolog, the promoter sequence of the A homolog might be different in some way than the B and the D homologs. But we saw no evidence for differences in promoter sequences being more divergent in cases where one of the 3 homologs was expressed at a different level. And we also didn't find any differences in transposable elements in the promoter sequences. So next we turn to look at epigenetic marks. And here this is showing H3K27 trimethylation across a balanced triad. So you'll have to believe me when I say there are actually 3 lines plotted, but they're basically superimposed. So the A, B, and D homolog have a very similar pattern of H3K27 methylation. When we looked in dominant triads, so for example, in this A dominant triad in the middle, we could see that the dominant homolog, in this case, for example, A, had much less of this H3K27 trimethylation than the non-dominant homologs, so the B and the D in the example above. And we saw the opposite pattern in the suppressed triads, where the suppressed triad has a higher level of H3K27 trimethylation than the non-suppressed homolog. And we also saw the opposite pattern in active histone marks, H3K36 trimethylation and H3K9 acetylation. And we also saw similar trends in gene body CG methylation, indicating there is a link between which homolog is more highly expressed and the epigenetic status of those genes, although we can't say whether the differences in epigenetic marks is driving expression level, or whether the expression level is driving differences in epigenetic marks at the moment. So we were curious how these differences came about during the evolution of polyploid wheat. And there was already some RNA-seq data available doing kind of the ideal test. So looking at a tetraploid wheat and a diploid wheat, crossing them together and then forming a newly synthesized synthetic hexaploid wheat that now contains the A, B, and D genome. So we could compare what would you expect to see in terms of expression level by adding up the tetraploid and diploid expression level and comparing to see how that really looks compared to what happens in the synthetic hexaploid wheat. And this is what happens. So in general, triads that were balanced in the ancestral species remain balanced in the synthetic hexaploid. So the A, B, and D copies had a similar expression level. Whereas in triads that were unbalanced to start with, such as A dominant and A suppressed, B dominant and B suppressed, or D dominant and D suppressed, these frequently changed their expression patterns between the ancestral species and this synthetic hexaploid. And in total, 47% of these unbalanced triads changed their expression pattern during the process of polyploidization. We then wanted to know what happened during the 10,000 years between becoming a polyploid and the wheat that we grow now on in farms around the world, and were differences in homolog expression also seen during this domestication process. So we couldn't really rerun the 10,000 years of evolution, but we could look at what were the differences between this newly formed hexaploid and existing wheat varieties as a proxy for what happened during domestication. And what we could see was that it was a similar trend again, that balanced triads tended to stay balanced between the synthetic hexaploid and the modern wheats, whereas unbalanced triads tended to change actually in this case. So 68% of triads changed in their expression pattern. So these results really suggested that there was significant changes in the transcriptional expression patterns in wheat. These happened immediately upon polyploidization, so from the ancestral species until the synthetic hexaploid, but also after polyploidization in these putative 10,000 years of evolution as a hexaploid, but also I would say that there were already pre-existing differences between the ancestral species. So it's not that all the genes, the homologs, were expressed at the same level, and suddenly upon becoming a polyploid, they all changed expression level. Some triads already had different expression levels. And these differences during evolution made us wonder, well, if this changed so much during 10,000 years, did it change the same way in all varieties, or actually are there differences in the expression levels between homologs in different varieties? And we compared between two different varieties, Chinese Spring and Azanaya, and we found that in 75% of triads, they had the same expression pattern in both varieties, whereas in 25% of triads, they had a different expression pattern in the two varieties, which might suggest there's some functional importance to differences in homolog expression patterns. And I was really curious what would happen if you cross together two varieties that have a triad with a different expression pattern. So for example, this triad in the blue variety has a higher expression from the B homolog, whereas the same triad in the orange variety has a higher expression level in the A homolog. What happens if we cross these together and look at the offspring? So we did this and looked at the F6 in a segregating F6 population to eliminate effects of heterozygosity. And we saw some offspring that had inherited the alleles from the blue parent, shown by the blue letters A, B, and D, and they had an expression pattern very similar to the blue parent in this triad, so that kind of made sense. We had some triads, some lines which had an expression of this triad that could be predicted by the allele that they inherited from the parent. So for example, in line 2, the A homolog has a reasonably high expression level. It's inherited the orange allele from the orange parent, and that's explained by what the orange parent's expression pattern looks like. And the B and the D homolog in line 2 are both inherited from the blue parent, and again, they follow a similar expression pattern to the blue parent. And the same is true for line 3. But we did see some cases where the offspring had a completely new expression pattern that couldn't be predicted by the alleles that was inherited. So for example, in line 4, the A homolog is really highly expressed, even though it's inherited the blue allele from the parent that didn't really express the A homolog. So this suggests that at some point, by crossing these varieties, at some point there's been a rebalancing of expression levels within this triad. And looking more globally across all the triads in the genome, the 11,000 triads, we can see that these triads are quite variable across these 47 different F6 lines. So some triads are always highly conserved. They look very similar to the parents, whereas some triads actually vary a lot and don't tend to keep the parental expression patterns. So we're now currently working on trying to understand what are the functions of these highly variable triads? Does it have any biological relevance? And also, what is the mechanism that determines which triads, if you like, maintain their homolog expression patterns after crossing, and which triads can be very variable in the offspring? So in conclusion, we found that in wheat, that 30% of triads show unbalanced homolog expression, which is interesting because this could be a potential way to improve wheat by only needing to worry about one or two of the homologs rather than needed to be concerned about all three homologs, as is the case for balanced triads. We found that homolog expression levels are correlated with epigenetic marks. We don't know which is cause and effect, but that's something that we're continuing to work on. We also found that homolog expression changed both during, immediately upon polyploidization and also after polyploidization. And we're now extending this to try and understand how differences in homolog expression pattern occur in different wheat varieties and what the functional significance of this is. And I'd like to end by thanking lots of people who've been involved in this project. I'd first like to thank Cristobal Wowie and Ricardo Ramirez Gonzalez at the John Innes Centre, with whom a lot of this work was done, and other members of the Wowie Lab who contributed to this work, including Sophie Harrington, Jemima Brinton, and Tobin Florio. I'd like to thank members of my lab at the University of Birmingham. So we're quite a new lab, we're just starting up. So hopefully you'll hear new stories from them quite soon. And I'd like to thank the International Wheat Genome Sequencing Consortium and all of the people listed here who were important in building the genome assembly and also providing annotations and RNA-seq data. And I'd like to thank all of my funders for supporting this project. And I'd just like to end by a quick plug. So I'm going to be hiring a postdoc to start working in this area in my lab at Birmingham. So if anyone knows anyone who will be interested in working on this topic, please put them in touch, and I'm hoping the start date will be in January. So thank you very much, and I'm happy to take any questions.
Thanks very much, Philippa. That's a great talk and a great opportunity if anyone wants to get a postdoc position there. So let me start with a couple of questions. So everyone, if you want to ask questions, just put them in the question box there. So first question I got from Ho-Han Go. This is a more of a technical question. He asks, how do you distinguish between when you do RNA-seq short reads, how do you distinguish between the different homologs?
Yeah, so that's a really good question, something we were also very worried about, because if you have mismapping between the three homologs, then all of these results might be nullified. So we've used the Callisto pseudo-aligner for our pipeline to detect homolog-specific expression, and this is has an intelligent algorithm, if you like, that can distinguish between the three homologs quite effectively, even in short reads. So the homologs on average are 97% similar. So in a short read of 100 base pairs, you would expect to have 3 base pairs different, and most of this RNA-seq data we used in this study was paired end, which also increases the chance that you can tell the difference between the homologs. The other thing we did as a control was we used null tetrasomic lines that exist for wheat, where a whole chromosome is missing. For example, chromosome 1A is completely missing from that wheat line, and carried out RNA-seq on that. And you would expect that genes on chromosome 1A shouldn't show any expression, and we found less than 1% of the wild type expression level on genes of chromosome 1A. So we're pretty confident that our expression levels are accurate between the different homologs.
Okay, cool. So a question from Godwin James. He asked about transposable elements. So he says, do you check the presence of TE elements in the proximal regions of specific homolog genes?
Yes, we did. So in terms of changing the expression patterns, we didn't see any differences in the content of transposable elements between the three homologs. Something I didn't have time to talk about today was differences between different tissues. And we did find some evidence that genes that the triad has changes in expression level between different tissues. So for example, the A genome might be highly expressed in the grain, but then the B genome is highly expressed in the leaves. We did see some slight differences in transposable element content then in the promoter sequences. So there might be some influence at some level of regulation of the homologs, but this picture is still not that clear.
Okay, so a question from Stuart Sullivan. He asks, for the variable triads in the F6 lines, did you find that they're enriched in a particular functional class of a protein or genes?
It's a good question, but I don't have the answer to that because that's still quite recent work. So yeah, it's still under investigation. So yeah, check back in I guess 6 months or so, and we'll have the answer to that hopefully.
Okay, so a question from, let's have a look, it's a long one. So a question from Amdeep Kaur. They ask about the triad studies again, and the results when we get the triad studies, do you think they can explain the results you get from conventional breeding with regard heterosis? So you see, when you get additional effects in the F1 and things.
It's a really interesting question. I don't have any data to say yes or no. My kind of gut feeling would be that probably in hybrid lines, you also get some interactions between the different homologs, and perhaps expression is, if you like, reshuffled in the way we see in F6 lines, but at the moment we don't know to what extent that is true. Yeah, but it's an interesting question for the future.
Yeah, absolutely. So some of the questions are quite long, so excuse me while I read them, work out. So this is a question from Dae Kwon Ko, and they say, in hybrids and allotetraploids, non-additive expression patterns has been known to be highly affected by environmental stresses. Have you looked at any environmental, the effects of any environment on these weeds expression?
The short answer is yes, and there's not a really clear pattern, so I think it will be different for different triads. So the data I've talked about today was all in control conditions, so no deliberate stresses applied. And in stress conditions, there are some triads which we know change in expression pattern, and looking at our data, we can see that, for example, in drought and heat, there are huge amounts of changes in gene expression pattern. I'm sure some of those will be changes to overall like triad homolog expression bias, but I don't have a kind of answer what the role is in stress tolerance. I think that's something that needs further exploration.
Okay, so a question from James Cockram, and he asks whether you've investigated target homolog expression in the F1. He says, as the genetic background is identical in all F1s between the two inbred parents, any differential expression of two alleles in the F1 would indicate cis versus trans control of expression.
Yes, that's a good question, and no, we haven't looked at the F1. So this was data generated by a previous study that we've reanalyzed. So unfortunately, they didn't look in the F1, but that's something we plan to do, hopefully with this postdoc, who I'm looking to hire. So yeah, watch this space.
You're thinking along the same lines. So another question from Stuart Sullivan, again, thinking about those F6 lines, and he asks if you see any phenotypic differences. I assume you do see quite a lot, but do you see any phenotypic differences, I guess, which go along with the difference of expression within the triads?
So we haven't specifically looked at phenotypic data in these F6 lines and tried to link it to the homolog expression patterns. So that would be something interesting to do in the future. I think at the moment we're still in the, I guess, quite preliminary discovery phase of trying to understand what's going on in these F6 lines and understanding how the homolog expression patterns change. And there's so many different traits you could imagine that it would influence. It's a bit difficult to know where to start, but that would definitely be something that's interesting to look into.
And cool. And then let's finish with a question from me about basically the stuff you said at the very start about that GW2 line. So you obviously showed 21% increase in seed weight in those lines, but do you see any negative effects in those lines, or is that all benefits? Do you see actually more productivity, or is it just more? Is there? Well, do you see?
So I have to say that isn't a study that I led. So I think from what I remember that it increases grain size but didn't increase yield, but I could well be wrong. So please check the paper for the real information about that. So
go back and have a look at it. Okay, that's great. So thanks very much, Philippa, for that. That's really interesting talk. And as I say, if anyone wants to get involved with it, then please do apply for that postdoc position. Thanks very much.
Okay, thank you.
Okay, so thanks very much. So let me get